Hello everyone and welcome to the Edupedia world. In the second session of the lecture on sexual reproduction in flowering plants, today we shall discuss about pollination, the common types, chief modes of pollination, importance of pollination, merits and demerits of the different types of pollination. In the previous lecture, we have thoroughly discussed about the structure of a flower, its various parts, the male and the female reproductive parts, the accessory parts of a flower, and also we have discussed about the variations in these structures. So now let's have a quick recap and move on to the next lesson. As previously discussed in this figure, we can see the structure of a complete flower or a perfect flower. Perfect flower is also called a bisexual or hermaphrodite flower. It consists of all the four whorls, that is calyx, corolla, androecium and gynoecium. Androecium is the male reproductive part and includes stamens. Stamens are in turn composed of anther, filament and connective. Here I shall not go into much details, shall just quickly recollect the terms and proceed on. You can also see in this figure the gynoecium, which is the female reproductive part of the flower. The gynoecium consists of carpels or pistils. The carpel is composed of three parts: stigma, style and ovary. Ovary contains the ovule. Besides the primary parts, there are accessory parts as well, which provides protection to flowers in the bud stage or help in pollination. These include sepals and petals. Previously, we have also discussed about the sexual stages in the life cycle of angiosperms that follows alternation of generations. The haploid gametophyte alternates with the diploid sporophyte during the sexual reproduction of angiosperms. The first structure in the haploid stage is the microspores. The microspores develop into the male gametophyte and are much smaller than the female spore. Pollen is the male gametophyte and develops in the anther. The other potential start of the haploid stage is the megaspore, which develops into the female gametophyte or the embryo sac. Once the gametophytes are fully developed, they produce male and female gametes. The male gamete is the sperm cell and the female gamete is the egg cell. The mature male gametophyte, that is the pollen grain, develops the sperm, while the mature female gametophyte develops the egg cell. Now, after a quick recap, let's begin with the topic of this session, that is pollination. So, what is pollination? It is simply defined as the transfer of pollen grains from the anther to the stigma of a flower. By now, you must be well aware of the different structures of a flower like anther, stigma, style, ovary, ovule, etc. Now, you must be wondering what is the need for pollination, or why pollination is important, or what role does pollination actually play in the life cycle of a plant. So, to all your questions, here lies the answer. Firstly, in the preceding session, we have learned that the male and female gametes in flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. So, how is this achieved? Pollination is the mechanism to achieve this objective. The male gamete, that is the sperm cell, and the female gamete, that is the egg cell, fuse together to form the endosperm nucleus in a process called fertilization. Secondly, it also helps in the production of seed. Seed is the final product of sexual reproduction and is described as a fertilized ovule. As we all know that all living organisms have one basic common goal, that is, to pass along the genetic information to next generation by creating offspring. Flowering plants create seeds which carry the genetic information of the parents and develop into a new plant. So, when pollen grains from the anther of one flower falls onto the stigma of a flower of another plant, it results in increase in genetic diversity because different flowers are sharing and mixing their genetic information to create unique offspring. Now, let's come to the different types of pollination. The two most common types of pollination include self-pollination and cross-pollination. Cross-pollination is also called xenogamy. Self-pollination is in turn of two types, autogamy and Geitonogamy. Now, coming to the two types of pollination in detail, first let's discuss about self-pollination. Self-pollination chiefly involves transfer of pollen grains from the anther to the stigma of the same flower. If the transfer of pollen grains occur from the anther to the stigma of the same flower, remember the term same flower, then the condition is called autogamy. However, if this transfer occurs to a different flower but of the same plant, remember same plant, then it is called Geitonogamy. After the self-pollination, let's move on to cross-pollination. Cross-pollination refers to the transfer of pollen grains from the anther to the stigma of a different flower of another plant. Now, moving on to the first type of self-pollination, that is autogamy. Here, pollen grains fall from the anther to the stigma of the same flower. Remember, autogamy is possible only when anther and stigma are close together and there is synchrony in pollen release and stigma receptivity. In cleistogamous flowers, that is closed flowers or flowers which do not open at all, the anther and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers invariably show autogamy as there is no chance of cross pollen landing on the stigma. There are several plants that show autogamy such as oxalis, balsam, rice, wheat, pea, etc. One more interesting thing to remember is that autogamy does not require any pollinating agent. In contrast, if the pollen grains from the anther of one flower lands onto the stigma of a different flower of the same plant or genetically similar plant, remember same or genetically similar, then the condition is called Geitonogamy. Unlike autogamy, Geitonogamy requires a pollinating agent. Although Geitonogamy is functionally cross-pollination involving a pollinating agent, genetically this is similar to autogamy since the pollen grains come from the same plant. In this figure, you can see a diagrammatic representation of autogamy, Geitonogamy and cross-pollination. Hopefully, the fundamentals are clear by now. Now, after learning the basics of self-pollination, that is autogamy and Geitonogamy, you must be wondering now how self-pollination is guaranteed or ensured in plants, or what enables self-pollination. So let's quickly review the devices that ensure self-pollination in plants. For self-pollination to occur, flowers are bisexual. That is, both male and female reproductive parts are present together in a flower. Also, both the sexes mature at the same time. That is, maturation of both anther and the stigma is synchronized. In some cases, like in Cleistogamous flowers, the anther and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma and there is no chance of cross-pollen landing on the stigma, thereby ensuring autogamy. Also, in some flowers, pollination occurs in bud condition, that is, before the opening of a flower. The opening of a flower is also called enthesis. Remember this term. Such terms are quite important, often asked now and then. Now, do you think is self-pollination advantageous to the plant? So yes, self-pollination indeed confers certain advantages to the plant. Firstly, it helps maintain genetic purity or parental characters indefinitely. Since, in case of self-pollination, pollen grains from the anther of one flower falls onto the stigma of the same flower or a different flower of the same plant or genetically similar plant. Therefore, it results in no mixing of genetic information and hence purity is maintained, unlike cross-pollination, which involves mixing of genetic information from two different plants to create unique offspring. Secondly, self-pollination does not require any pollinating agent. Therefore, plants do not spend extra energy in the production of pollination attractants like producing brightly colored large petals, nectar, scent, etc. Also, since the pollen grains are not carried away far off places, therefore pollen grains need not be produced in large numbers. Again, makes the process economical. But like the two sides of a same coin, it has several disadvantages as well. Since self-pollination does not involve mixing of characters or genetic information, no new species are produced or no unique offspring are produced with unique set of traits. Instead, parental traits are maintained in successive generations, and also continuous self-pollination through successive generations lead to reduced vigor and vitality in the progeny, resulting in reduced fitness in offspring. Since no new characters are introduced in the resulting offspring, no variation occurs in characters. Hence, it regresses the chances of evolution. As a result of self-pollination, undesirable recessive traits are also not eliminated and they propagate through successive generations. Immunity to diseases also decreases in case of self-pollinated plants. After discussing self-pollination, let's begin with the fine details of cross-pollination. As mentioned earlier, unlike self-pollination, cross-pollination basically involves transfer of pollen grains from the anther to the stigma of a different flower. As in the figure, you can see pollen grains from stamens sticks to a bee as it visits a flower. The bee travels to another plant of the same type. The pollen on the bee sticks to the pistil of a flower on the other plant and brings about cross-pollination. Now, you must be wondering whether this process is advantageous to the plant. So, yes, this process is indeed highly advantageous. This process involves two different plants and thus mixing of characters from two different plants, unlike self-pollination, occurs, which ultimately results in the exchange of genetic information and production of offspring with unique characters or new varieties of plant. The resulting progeny also shows enhanced vigor and vitality. The offspring produced are more viable as they show greater degree of fitness and exhibit resistance to plethora of diseases. Cross-pollination is an essential part of sexual reproduction in seed-producing plants. It allows for genetic recombination or shuffling of genetic material between two different plants and formation of genetically unique seeds. It also enables production of seeds in self-sterile plants. Since this type of pollination involves mixing of characters from two different plants, it enables introduction of new traits in offspring, increasing variation and also elimination of undesirable recessive traits from the population. Despite several advantages of cross-pollination, there are certain disadvantages too. Since the pollen grains are carried to far-off places, pollens are produced in large numbers and often involve wastage. Unlike self-pollination, pollinating agents are required to transfer pollen grains from the anther to the stigma. Plants also develop several devices to attract pollinating agents, like they produce scent, nectar, petals are brightly colored, large, showy, etc. Cross-pollination is not at all an economical method. If cross-pollination has to occur, pollen needs to be carried or transferred from one flower to another flower of a different plant. So, now you must be wondering how this is accomplished, as the pollen grains are non-motile. What are the agents of cross-pollination? So, the two broad categories of cross-pollination agents include abiotic and biotic agents. Under abiotic agents, we chiefly have wind and water. Wind-mediated pollination is called Anemophily, while water-mediated pollination is called Hydrophily. Remember these terms. Amongst biotic agents, we have insects. The process is Entomophily. We have birds, that is Ornithophily, bats, Chiropterophily, and ants, Myrmycophily. Now, let's discuss about the peculiar characteristics or adaptations in flowers that facilitate pollination by wind. For instance, plants that show wind pollination do not have large, brightly colored petals. Instead, they have small petals, often brown or dull green. Since such plants do not attract insects for pollination, they do not produce scent or nectar. But such plants do produce pollen grains in abundance because it has to be carried off to far off places, and most of them fail to reach distant flowers. Since the pollen grains are carried away by wind, they are very light and smooth. Further, to cause easy release of pollen and carried by wind, the anthers are loosely attached and often dangle out. In order to catch the drifting pollen, the stigma hangs outside the flower. Also, in order to catch the drifting pollen swiftly, the stigma is feathery or net-like. After wind pollination comes the pollination by water, also called hydrophily. Hydrophily is in turn of two types, epihydrophily and hypohydrophily. The term epihydrophily implies pollination taking place above the water surface. The prefix epi means above, and hydrophily refers to attraction or affinity to water. Hydro is water and phily is affinity or attraction. Vallisneria commonly shows epihydrophily. On the other hand, hypohydrophily refers to pollination taking place below the water surface. Plants like Zostera, Ceratophyllum show hypohydrophily. Now, in order to facilitate pollination by water, plants must possess certain adaptations. For instance, pollen grains are lightweight and covered with wax. Here also, the flowers are not brightly colored and do not produce scent and nectar. The stigma is long and sticky. Pollen grains are produced in large numbers to prevent loss. Flowers are generally unisexual. After the abiotic modes of pollination, let's come to the biotic modes of pollination. Amongst the several biotic modes as mentioned in the preceding slides, let's first discuss pollination by insects, commonly called Entomophily. Here, unlike Anemophily and Hydrophily, the petals are brightly colored, large and showy, are often sweetly scented and contain nectar to attract insects for pollination. Pollen grains are also produced in moderate quantity since it involves less wastage than pollination by wind or water. Pollen grains are often sticky or spiky to stick to insects. Anthers are firm and inside the flowers with stigma. Stigma has sticky coating so that pollen sticks to it. Pollination by birds also involves certain adaptations like flowers are large and brightly colored, they produce mucilaginous nectar, pollen grains are sticky and flowers produce fleshy and thick floral parts. Flowers generally do not produce scent in this case. Plants like bombyx, bignonia, bottlebrush, etc. show ornithophily. While pollination by bats involve adaptations like flowers open during evening or night. Flowers are large, dull colored and have a strong scent like that of rotting fruits. Besides, flowers also produce large number of pollen grains. Flowers are also tough and produce nectar. Plants like Kigelia anthocephalus show pollination by bats. After discussing the two basic types of pollination, that is self-pollination and cross-pollination, its several advantages and disadvantages, we shall now discuss another interesting point called outbreeding devices. What are outbreeding devices? As we have studied earlier, that majority of plants produce hermaphrodite flowers and undergo autogamy. However, this continued autogamy or self-pollination results in inbreeding depression. Therefore, plants have developed several devices to avoid self-pollination and encourage cross-pollination. Such devices are called outbreeding devices. For instance, in some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the pollen release. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices check self-pollination. The third device to prevent inbreeding is self-incompatibility. Now, how do you define self-incompatibility? Self-incompatibility is chiefly a genetic mechanism that prevents self-pollen, that is, pollen from the same flower or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, it prevents autogamy but not Geitonogamy. In several species such as papaya, male and female flowers are present on different plants, that is, each plant is either male or female. This condition prevents both autogamy and Geitonogamy. Now, let's move on to another interesting point, pollen-pistil interaction and its role. Pollen-Pistil interaction primarily comprises series of events from the time of pollen deposition over the stigma to the time of pollen tube entry into the ovule. In this figure, you can see the deposition of pollen grains on the stigma and the course of the growth of pollen tube into the ovule. Actually, what happens? Pollination does not guarantee the transfer of the right type of pollen. Often pollen of the wrong type, that either from other species or from the same plant, if it is self-incompatible, also land on the stigma. In that case, the pistil has the ability to recognize the pollen, whether it is compatible or incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that lead to fertilization. However, if the pollen is of the wrong type, that is incompatible, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. And this ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil that is mediated by chemical components of the pollen interacting with those of the pistil. So, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. You would recall that in some plants, pollen grains are shed at two-celled condition, that is, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. This filiform apparatus present at the micropylar part of the synergids guides the entry of the pollen tube. So, all these events from pollen deposition on the stigma until pollen tubes enter the ovule are brought together as pollen-pistil interaction. Now, after understanding the basic concept of pollen-pistil interaction, you must be now convinced to agree upon the fact that this interaction between the pollen and the pistil is a highly dynamic process and might help the plant breeder in manipulating pollen-pistil interaction even in incompatible pollinations to get desired hybrids. So with this, we come to an end of this session on pollination. Till now, we have discussed various types of pollination, their advantages and disadvantages, the outbreeding devices, the fundamental concept and the importance of pollen-pistil interaction. In our next lecture on sexual reproduction in flowering plants, we shall discuss about double fertilization and different post-fertilization events. Thank you for watching this video. Feel free to catch up with me with your queries, if any, regarding these sessions. Hope to see you again.